Hello, everyone, and welcome to the Global PKU Conference. My name is Thomas Christiani, and I'm a scientist working on a home monitoring platform called the PKU Now, which can quantify phenylalanine concentration directly from a finger or a heel stick blood sample. Our company, In Vitro Diagnostic Solutions, was first founded by Robert Harper in 2017. We focused on the development of novel point-of-care diagnostic devices for PKU, as well as other metabolic disorders, such as glucose 6-phosphate dehydrogenase deficiency, jaundice, which is related to the bilirubin content found in your blood, urea cycle disorder, and maple syrup urine disease. Our team has raised over a total of $7 million since our inception of non-diluted funding from both the National Institutes of Health and the National Science Foundation. IVDS has also contributed to a total of 10 products on the market, 8 patent applications, and 16 successful SBIR grants. Currently, we are developing a universal platform that will help us leverage our test strip and handheld meter across multiple diseases, including PKU. As most of you are aware, PKU is an incurable genetic disorder characterized by the buildup of phenylalanine in the blood, which can then lead to severe intellectual disabilities, seizures, behavioral abnormalities, microcephaly, as well as skin diseases. There have been many advances in the treatment strategies for PKU. These include a variety of food supplements in both liquid and powder form, and these are manufactured by several companies as shown on the slide here, such as Nestle, Cambrook, Mead Johnson, and Nutricia. BioMarin has also developed 2 types of medications, both oral and injectable, which aid in the reduction of phenylalanine levels in blood. Today, the area of diagnostic technologies can be greatly improved, and this is where we feel that IVDS can really make an impact on the PKU patient population. Despite all this progress, there is still an unmet need to provide daily real-time blood Phe results. As patients are aware, the cost of testing remains high, and results can take several days to process. Real-time decisions cannot be made due to this delay in feedback. In addition, results can be inaccurate due to the low quality of testing, or additionally, the interference of tyrosine when using spectrophotometric methods. Therefore, an at-home point-of-care diagnostic tool would dramatically change the way in which PKU patients can monitor their blood Phe levels. Our solution to this is the PKU platform. The PKU platform is a cost-effective kit that contains a finger and heel stick, disposable capillary transfer pipette, a capillary stand to hold the pipette for ease of obtaining blood, test strips, and a handheld meter. The meter has Bluetooth connectivity and can store and disseminate results, diet, and medication dosage remotely. Each file has a QR code which provides the test strip lot number, curve set, and expiration date of the test strips. The meter utilizes 3 light-emitting diodes that can simultaneously measure 3 different analytes in blood. In our current platform, we use the red LED to measure Phe concentration in the reagent layer of our test strip. The PKU test strip architecture allows for the filtration of red blood cells from plasma in situ. Plasma reaches the PKU detection pad, and the measured end color of the reagent layer is proportional to the blood Phe concentration. The PKU now has a broad analytical range from 1 to 25 milligrams per deciliter. Results are then displayed on the cell phone and can be stored, archived, and disseminated via an encrypted HIPAA-compliant cloud-based platform. To verify the accuracy of the PKU Now, de-identified whole blood samples were spiked with phenylalanine across the analytical range and tested. Plasma from these spike samples was harvested and sent to the University of Wisconsin for analysis using HPLC, amino acid analysis, and tandem mass spec. The data reveals excellent agreement between measurements taken with the PKU Now and both HPLC and mass spec. As you can see in the bland Altman plot of 60 determinations, it illustrates that nearly 97% of the data points exist within 2 standard deviations around the mean. This indicates that there is good agreement between the PKU Now and HPLC results. Additionally, we spiked 3 different samples at concentrations of 1, 3, and 6 milligrams per deciliter of Phe, as well as spiking the samples with tyrosine at a concentration of 6 milligrams per deciliter. High levels of tyrosine have been known to interfere with the low levels of phenylalanine and can therefore create a false positive or read as above normal levels for phenylalanine. Our design can actually suppress this interaction between tyrosine and phenylalanine dehydrogenase and prevents infidelity of the enzyme with Phe. In order for the FDA to categorize the PKU now as a CLIA wave test, IVDS must demonstrate that the device is simple to use, exhibits low risk to the public health, and an untrained person can obtain reliable and accurate results based on a set of instructions. Therefore, 2 synthetic quality control solutions containing 5 and 14 milligrams per deciliter of Phe were evaluated using the PKU now. Our results indicate that the spiked values align with the control samples. Shown here is a projected timeline for the development of the PKU now technology. Due to the support of the NIH and general medicine, the PKU now home monitoring meter is on track to be available on the market by approximately early 2023. In summary, the PKU now will dramatically change the way patients with PKU can monitor and track their blood Phe concentration. Real-time results will be readily available and will only require a low blood volume from a finger or heel stick. Our technology is sensitive, cost-effective, and will be able to easily manage and store data via Bluetooth on a cell phone. IVDS has obtained $1.4 million from the NIH to complete development efforts and lock in a final design. Once we certify, verify, and validate the PKU now, we will move on to clinical trials with our collaborators from UPMC. We are also aiming to further develop a test strip with increased sensitivity and resolution at lower Phe concentrations within the range of 0 to 4 milligrams per deciliter. IVDS is currently seeking a licensing partner for additional clinical studies and FDA submission. Our team would like to thank our collaborators, funding sources, and the Global Association for PKU for allowing us the opportunity to share the development of our work. Thank you.